Um, I want to say at the outset that this is both a research project and a monitoring project. We are, collecting data that should help decision makers to better protect the students, but we're also learning a lot as we go. And although this has been done for some time now, there's still a lot of unanswered questions which we're working on. So the basic idea is we're collecting wastewater samples and analyzing it to look for the COVID virus. And if we find it, that's a good indication that all of that, somewhere in that water that ended up in the sewer, someone is infected. And not only can we just look for it, but with some of the sophisticated techniques that Karen is using, we can get an idea of how much COVID is present and how much infection is in the population there. All right, so right now, the university has taken a very aggressive stance to protect the students. They have a very vigorous testing program in place. All the on-campus students are tested weekly, and that's really kind of the gold standard. That's, if you want to monitor how the infection is spreading through the community, the best thing you can do is test everyone as frequently as possible. So what we're doing here is quite a bit different, and it provides a little different information for them to make use of. So when someone gets infected with COVID, people respond to it differently. So once you get the infection, it can take up to 2 weeks before you exhibit symptoms. So, for that 2-week period, you could be infected and infecting other people and unaware of that until you start to feel sick. The other strange thing that can happen is there's a lot of there are asymptomatic people out there. So you get infected and you never get sick, and you can also be infecting other people without knowing it. Once you get infected, you shed virus. So you release this signal out into the environment that you have COVID. And that's what Karen and I are really looking for. We're trying to make use of that sort of invisible signal that's out there. Now I'm going to get a little graphic here, but you guys all read the title of this talk, so there shouldn't be any surprises here. But basically, once someone is infected, the COVID virus is in their feces, and when they go to the bathroom and flush the toilet, that COVID virus gets sent down the toilet into the sewer. And then we can collect a water sample and then separate the virus from all the other junk that's in there and look for that and determine how much virus is present. So like I said, this is a crazy way of doing this, right? But it does have its advantages. Like, for one, it's a composite sample. So when I collect a sewer sample, it is all of the water that came from an entire dorm. So it's really representative of everybody living in that dorm. So with one sample, you can get an idea of how bad the illness is in that dorm. And that can be really useful. It's a lot cheaper than testing each individual in the dorm. And since it is a community, you get a sense of to what's going on in that individual dorm. And it can be useful for decision making. The other thing that is useful with this is that, when I collect a sample and send it to Karen, she can get and post results within 12 hours. That's way faster than waiting for people to get sick. So, if you don't have a vigorous testing program in place, you can catch the disease spread before you see illness in the population. And then finally, for OU, I think this is a useful aspect to it, is that when you have individual testing, it's it's a burden on the individuals, right? Like every student every week has to schedule an appointment and go and get tested. And, we make it as easy as possible, but still it's one more thing to do, right? And we've been at this for a year now. Everyone's a little weary of the pandemic. And you always have a certain level of non-compliance. So there's a certain subset of students out there that even though the test is available, they're not getting it. With the wastewater sampling, we're going to find them, right? We don't need compliance. We don't put the burden on the individual. We can just collect the water that's in the sewer. Okay, so I'm out here at one of the sewer locations. I'm going to head over there right now. One of the things I love about being a civil engineer is that we spend our days working outside. Some days that's awesome, but today it's raining and windy, so there's good and bad in that. I have employed a team. Whoops. There we go. I've got a team of 6 undergraduate researchers working with me right now, and these are 2 of the lucky individuals here today. Can you guys introduce yourselves? Just kind of yell it out. Grace?
Hi, I'm Grace. I'm a senior.
Hi, I'm Nate. I'm the senior as well.
All right. So you can see right off the bat, they are in extensive PPE to protect from infection. So anytime you're working with wastewater, and not just during COVID, but anytime at all, you run the risk of being exposed to waterborne diseases. So we want to prevent exposure to any of the liquid that's down in there. All right, Nate, let's get started and see what we can find. So you can see there's a manhole cover here. Took us weeks to find the right tool to get these things open. This is the 3rd tool that we figured out. But I just want to draw your attention to this amazing infrastructure we have. So here's a hole that connects to a pipe. Every drain in this building in front of me drains down into that hole. And then it goes from there to another pipe that goes to the wastewater treatment plant. So there's this incredible invisible infrastructure that we rely on every day to get rid of wastewater, to get our clean water, to get rid of stormwater off the streets. And I don't know, I'm being a civil engineer now, trying to get everyone to appreciate it because it is invisible and it's something we take for granted. All right, so you can see that when we do this sampling, we need this special little robot called an auto sampler. And the problem that we encounter is that we need to get a representative sample. One of the limitations of this technique is that if I just grabbed a sample right now, that would just be representative of what's coming down the pipe over those last 15 minutes, right? So what if someone didn't flush the toilet right then? And then also, I really want to get a composite sample of the entire dorm, right? So, but everyone doesn't use the toilet at the same time. So what this little robot allows us to do is we program it, and it collects a little bit of sample all day long and then puts it all together in a single bottle. All right, Grace, let's see what we can find here. Now, this is the most dangerous part of my talk. I don't want to lose my phone. So you guys may all end up down at the bottom here, and it'll be the end of my portion of the talk. So I don't know if you can see that, but that's what's down a manhole. And, I was talking about the infrastructure. So not only do we have to, connect all of these things to get rid of our waste, we also need to have ways to get in there and service it. So a manhole is literally a hole that's big enough for a man to fit down into, so they can go down there and clear things out when you have problems. And in this manhole, you can see there's 3 pipes. So there's 2 different dorms coming in here, or pipes from 2 different buildings coming in, and they're combining and going out to another pipe off to the right, and that goes to the wastewater treatment plant. And I don't know, I don't know what I expected when I first looked down here. I expected it to be a little more gruesome, I guess. but there is a lot of other water in here. So basically every drain from these buildings ends up down in this sewer. So sinks, showers, and toilets, all of that water all gets put together in the same pipe, gets sent down here, and then goes to the wastewater treatment plant.
Guy,
Right.
I have a question for you.
Yeah, fire away.
So one of the viewers wants to know is what we are seeing in the water, toilet paper.
Yep. Yep, that's exactly what it is. And it's already breaking down.
Thank you.
Okay, so you can see a tube coming from our auto sampler and going down into the water. At the bottom of that tube is a screen, and that's where we collect our sample. This manhole is about 8 feet deep, but some of them are as deep as far as 20 feet deep in our system. And you'll also notice that this is not pressurized flow. The water's just flowing in a channel at the bottom there. And that's how these sewers are set up. They're all gravity flow, so they have to be designed so that water just flows from all of the buildings downhill to the wastewater treatment plant. And if you ever tour the wastewater treatment plant, which I encourage, the water comes in there about 40 or 50 feet underground because it's got to be that deep to get gravity flow.
Guy, I have another question for you.
Yeah, go ahead.
It's the question I think you were expecting. Does it smell?
It's a really windy day, so we're good right now. But yeah, it can smell. We've had, once we opened one up, and a whole bunch of cockroaches came crawling out, which was really pleasant. Haven't seen any rats yet, but I expect it's just a matter of time. Yeah.
No fish either, I take it.
All right, so this is our auto sampler. And like I said, so we started this yesterday morning. I can't really read the display, but it should be on its 8th sample. So I should really have a student do this. I'm probably gonna mess it up, but let me see if I can figure this out. Oh, there we go. Yeah, it's on sample 10 right now. I'm gonna turn it off. I'm gonna try to, and then run it again. All right, hang on. Getting there. Oh, whoops. Okay. So what this does, here we go. Okay. I'm going to turn this on, and what it'll do is it'll collect the first sample. If I can do this right. Is it that button? All right. And you can hear the pump turning on. And what it's doing now is it's purging air out of the line down to the sewer. So it's just making sure that the tube is clear. So the pump was reversed. Now it's going to suck water up through the tube. And you'll notice the tube is quite long. So it's got to calculate how much water it's going to pull in. And then it just grabbed a little bit for its sample. And then it reversed, and now it's pumping the water back out again. Okay, so that's the whole cycle. So it clears the line, it pulls water up, and it knows how much water to pull to fill that amount of tube. And then it grabs a little bit more. It's also got a water sensor, so it knows when the water gets there. And then it adds that little sample to a bottle. And then it waits. And then when the program turns on again, it'll collect a little bit more. And it'll do that through a 24-hour period. All right, you guys take it apart, and we'll grab a sample. So Nate was here. When were you here? This morning?
Okay.
What's that?
What'd you say?
Were you here this morning? Yeah, so Nate and Grace were here this morning to set this up. At 8 o'clock, they started this and turned it on. So it's been collecting samples since 8 o'clock this morning, or 7:30 this morning. And so that's where the bottle is. The other thing about getting a representative sample, go ahead, Grace, is you can see we've got ice packs around it. We want to make sure that the sample that we grab is, that the sample that we grab is, has active COVID in it, and that by the time it gets to Karen and she gets to analyze it, we haven't lost any of that COVID. The best way to preserve it, the best way to do that is to keep it cold, and that slows down all the reactions and keeps the COVID active. All right. And
I'll have another question for you when you're ready.
Okay. Yeah. Let me just one more thing. So that's basically the process, and that's basically my contribution at this point. That's what we deliver to Karen to do her magic. Okay, Roxanne, shoot.
It's a multi-part question. The first part is, how many sewers are you monitoring?
5.
And are they all on campus and dorms?
Yes. So right now, the funding that we have covers, and this funding comes through the state from the governor's office originally, and it covers 5 dorms on campus. And then we also have another proposal that's out there right now to start monitoring some locations in the city of Athens, but we're waiting to hear back if that gets funded.
Okay, just a point of clarification. Are you monitoring, 5 dorms or 5 sewers?
Oh, yeah, good question. So we're monitoring 5 sewer locations, and each of those comprises anywhere from 1 to 3 different dorms. So some of them, the water sample we're collecting is a composite of 3 dorms. So I think anywhere from 50 to about 300 students in each sewer location.
Okay, and how often are you collecting those composite samples?
Twice a week. So the students come out here Sunday and Wednesday morning to set up the auto sampler, put the bottles on ice and turn it on. And they come back Monday and Thursday morning to collect the sample and deliver it to Karen.
Thank you.
All right, so Karen, if you're ready, now it's your turn. So normally the students, normally the students have to put these bottles in a cooler on ice and then deliver it to the refrigerator in Karen's office. But we're going to take advantage of the technology we have here, and you got it?
Thank you. Yes.
All right, there we go. We should do that from now on.
Hi, much easier. Well, what I'm going to tell you about is the molecular aspect of how we monitor for the virus in this wastewater. And I have to say, Guy's team, they are phenomenal. Rain, sleet, snow, hail, they are out there like clockwork, and they are delivering the samples to me like clockwork. So I don't have any snow days because they don't take any snow days. So I just want to show you, I'm going to share some slides with you. And so this is what the wastewater samples look like, or can look like. On one side, I've shown where they're settled out. They've been sitting for a while, but the other side shows where they've been shaken. And you can see they're still, even though there's a screen on the end and for when the wastewater's sucked in, brought in, a lot of debris still comes in. And I'm not quite sure what all this debris is. I know there's got to be some toilet paper fragments because I can kind of see that, but I don't know what some of the rest of it is. But before I actually tell you about the assays that I do, I wanted to tell you just a little bit more about the virus itself. And so this is just a stylistic picture of a SARS-CoV-2 virus. So SARS-CoV-2 actually stands for Severe Acute Respiratory Syndrome Related Coronavirus 2. And so that's the SARS-CoV-2. That's the name of the virus. And then COVID is actually, or COVID-19 is actually the name of the disease that results from infection with the virus, with the SARS-CoV-2. And that the COVID comes from coronavirus, so CO, disease. So corona, CO, virus, VI, and then disease, COVID, and then 19, because this was all first detected in 2019 in China. That's where this all was first recognized. So the virus, as I said, this is a stylized picture, but I wanted to show it to you so that you could have an idea, because you may have heard of some of the things that the assays are monitoring. For instance, you might have heard of the spike protein. And the spike protein, that's indicated, I don't know if you can see my mouse or not. The spike protein are these big red projections coming out of the virus. And these spike proteins are actually the first contact that the virus makes with a cell. So when it's going to infect, it interacts with the cell through these spike proteins and binds to a receptor on the cell. And that receptor is an ACE2 protein. So, and that we actually hear about through because of coronary disease and such. So, so that was one thing I wanted to point out, the spike protein. And I'll tell you a little bit more about some of the tests that target that. Another thing that, well, so this shows those spike proteins. Then we have like an envelope around. These viruses are called enveloped viruses. So they have a membrane around the outside. So there's a membrane protein, and there's also an envelope protein. And these are things that the virus actually encodes. The virus cannot make these things themselves. Viruses aren't actually living organisms, not in the sense of the word. They don't make energy. They don't metabolize. They're not living in that sense. What they do is they infect, and then they kind of hijack the host, the machinery in the host, and get that host machinery to translate the RNA. So SARS-CoV-2 is an RNA virus, and that's kind of shown here, depicted here with these squiggles. So the RNA, ribonucleic acid, is what kind of tells how to make these various proteins. So for instance, we have RNA in us, and we also have DNA. So our sort of the blueprint within all of our cells is what we call DNA in our cells, in human cells. And that DNA, deoxyribonucleic acid, is sort of like a blueprint that tells like everything that could be made within that cell. Yeah, everything. And that's going to be the same in all of our cells, that blueprint, that DNA. The RNA acts sort of as a middleman. It's a go-between between the DNA and then the proteins. So for instance, these red things sticking out or these white things. The proteins are kind of the workers. They do things. And so the RNA serves as that middleman, at least for us. Well, viruses, this type of virus, this specific virus doesn't have DNA, it has RNA, just has that middleman. And so what it will do when it infects you is it will inject that RNA, or the RNA will become, it will be in your cell. It won't integrate. It never integrates. It never becomes a part of you in that sense. But it will be in your cells, and then your machinery that would normally make those proteins will make proteins from the viral RNA, and so make viral proteins. And then those cells can reform more virus particles. So one comes in, many more go out. And so that's kind of the life cycle of the virus. And so the spike protein is important. We've got this membrane protein and envelope protein. We've got this lipid bilayer. That becomes important when I'll talk briefly about the vaccines at the end. And so that lipid bilayer is important. And then for my assay, this nucleocapsid protein gene, the gene for it, the nucleocapsid protein, this N gene, that's actually what I assay in my molecular assays of the wastewater. And so the nucleocapsid protein actually coats the RNA and helps package it inside the virus particle. I'm not assaying the protein, I'm assaying the sequence in the RNA that would encode that protein. Okay, so when we're working with this coronavirus, the SARS-CoV-2 it's contagious. We know that. Fortunately, with the virus that's found in the wastewater actually hasn't been found to be terribly infectious. We think that probably what's being shed is mainly inactive virus, or it's become inactive. It's not dead per se, but kind of. It can't infect. But we're not completely positive of that, so we use special personal protective equipment when we're working with this virus. So for instance, in the lab, this is a picture of me, my normal lab attire. I've got a lab coat, I've got gloves, just a single pair of gloves. I have my mask. That's just because of COVID right now. I don't normally have to wear my mask. But when I'm working with this wastewater, I have to put on more stuff. I have goggles that I have to protect my eyes. You can't see it, but I actually have an N95 mask underneath the blue surgical mask. And then I have an isolation gown. So garb that would normally be worn like in an operating room or such. So I have to wear this. So this is because at one stage I will be concentrating the virus, and that's where we're nervous that it could be a biosafety issue. Just in the wastewater, just hanging out in the wastewater, there's really not a large enough load, enough just free in the wastewater to really be of too much concern. Although you notice Guy's students, they still all dress up as well. We don't want to risk anything. But we in the lab will then concentrate it and try to pull it out and try to reduce the volume that we're working with. And over here on the left is a picture of my grad student, Saban Kadji, and he is helping me with these assays. He's helping me isolate the RNA, the virus, and then the RNA from the wastewater, and then do the assays later. And this just shows a biosafety cabinet. We do not open the wastewater bottles out in the open. We only open them inside that hood. And this over here shows the pelleted virus. So I start with 5 mils of the wastewater. So I'm only sampling 5 mils. I do it 3 times. And I have a special solution that I add that will help pull that virus out of the water. And when I spin it really fast in this little round thing down here, this centrifuge, the rotor of the centrifuge, the virus will, and also debris, that's really what you're seeing. You won't see the virus itself, it's too tiny, but you'll see the debris in the water will smack up against the side of the tube. And so that's the pelleted virus and debris. And so I will pour off the wastewater that's left. I pour that away, get rid of that. And then I'll resuspend, I'll pull that pelleted stuff off the wall, and then I transfer that into this tube over here. I call it my bead beading tube. It has little glass beads, tiny glass beads, and then I put it on a machine that will shake it like crazy. And so those beads will help break up the virus. And also some of the liquid, I put in a special liquid that will help lice break open the virus. So, and eventually there's various steps. I'm not going to show you those, but it's all molecular biology. In the end, we end up with RNA, ribonucleic acid. And so then I take that from this lab to a different lab where I do what I call one-step multiplex RT-qPCR, so reverse transcription quantitative PCR. And it's called one step because everything is done in just a single tube. So I put in, and I make this mix that has all of this stuff in it. It's got buffers and it's got enzymes. There's an enzyme for doing this reverse transcription. So it's making a copy of the RNA. And then I have a DNA polymerase. That's another enzyme which will make lots of copies of that first copy. And I have what I call sequence-specific primers and probes. So I'm actually looking for evidence of 3 different genes. So I look for the viral RNA, so evidence of the virus, from 2 different ways. I call it N1 and N2, and you might have heard of these terms. The CDC actually pioneered the assay for this and developed the sequences, the primers and the probes, so that we can do these assays. And so I've been using what the CDC developed for this part. So N1 and N2. So when I detect these 2, I know that I've got viral particles in there because I can detect the viral RNA. And you notice I'm color coding them here because what I'm doing, I'm calling this multiplex because I'm actually assaying all 4 of these genes, I'll tell you, or regions, all at the same time in one single tube. So I have to have a way of dividing them out. And so they have these fluorescent labels put onto them that will show a different color. And so I've colored, just for convenience, I've colored the N1 as blue, the N2 as green. And then because I'm trying to pull the RNA out of this wastewater, I don't know how well my method really works, and nothing's ever 100%. So before I even isolate that RNA, I spike in some bovine coronavirus. And I get this from a vaccine. Vets vaccinate calves, baby cows, against the bovine coronavirus. Well, we can use that vaccine as a spike in, we call it. We put that in a known amount into the wastewater, and I'll know what signal I put in, and then I can know what signal I get back, and I can calculate what kind of efficiency I've got. And then the last one I assay for is, we call it PMOV. I think it's a pepper mottled mosaic virus. And this is a virus that we all have in us. Pretty much everybody does. I think it mainly comes from eating peppers. I mean, everyone eats peppers pretty much. And so what has been found is that it ends up in your feces. And so this helps me determine how much feces was in that water that was collected. Because, we don't know how many people actually went to the bathroom during that 24-hour period. And we also don't know how many people, washed their hands, took showers, all these other ways of getting water in there and diluting it out. And so the PMOV signal will also give me an idea of how much feces was in there in the first place. And so how much virus could have been there. It gives me a sense of how many people were contributing. I can't name exact numbers of people, but it gives me an idea. So I put all of those together into this tube, and I put the RNA that I isolated from the wastewater into this tube. Now, it's actually not into a nice little tube like this. I'm working with this plate. You see these little bitty wells? That's, each one of these is what I consider a tube. There's 384 of these. And so we're pipetting into each of these little bitty wells. And so I go a little cross-eyed, but once I get my assay set up, I put it into this machine. And an hour and a half later, I get this. So isn't that beautiful? This is the fun part, the curves. You notice these are color-coded too. So the colors are showing which genes, which sequences I'm looking for. So these blue ones and the green ones would be indicating that there's virus there. The red one is my spike-in, my bovine coronavirus spike-in. And then the purple is that PMOV, so the feces indicator. And I don't really look at them all at once like this. I will, there's various ways I look at it, but this is showing just one sample. When I do my, the molecular analysis, I always do them in replicates of 6. I repeat them 6 times. And I've also extracted RNA from 3 wastewater samples. So really, I've got 18 samples, all reflecting that one sample of water coming from that one manhole. And
Karen,
so,
you have a
yes.
question, if
Sure.
I may. It says, since all these samples could be infectious, do you need to use a BSL-3 or a BSL-2 laboratory?
Very good. So this, the hood that I showed you a while back, I forgot to say, was a BSL-2. Because we aren't trying to grow the virus, we aren't trying to culture it and make more of it, we can work in a BSL-2 hood. If we were trying to grow it, then that requires BSL-3, and we do not have that capability on campus. And so we
just
do not
clarify, BSL is Biosafety Lab, correct?
level.
Or
Yeah,
level,
biosafety
excuse me.
level. Yep.
Thank you.
So most of what I work with is biosafety level 1. So you need gloves and such, but not too dangerous. Biosafety level 2, a little more because it could be infectious. Biosafety level 3 and 4. 4 is the highest level. Those you've got to be very careful. Like Ebola virus, I think that would be level 4.
Thank you.
So we work BSL-2 at least until it gets into that beading tube. And once it's in there, actually even before that, when we're precipitating it, it's been shown that the chemical that I'm adding, the liquid that I'm adding to precipitate the virus actually inactivates it. And also the solution I'm adding to release the RNA, that also inactivates it. So all along the way, I'm doing things that are going to inactivate that virus. So the PPE that we're using is more a precaution than a real necessity. And also, we're pretty sure that what's coming down the sewer system in the first place, so far, no one's really been able to grow anything from that. So no one's been able to actually culture and grow the virus. Not that we would try, but others have tried that in where they have the facilities. So we don't think that what's coming in the feces is actually active. And so it's unlikely to be able to spread and infect other people via that route. Okay, so.
One question, if I may. So it asks you mentioned that it's about an hour and a half to get these samples, but that wasn't from the very beginning.
True.
What they want to know is, with all these time points and all these samples, how long does it really take you to analyze a sample for each day that you do it?
So Guy's team, they collect the wastewater. So, the auto sampler, that machine has been collecting it over a 24-hour period. And then it takes them about an hour and a half to go around and collect those 5 bottles and then bring them to my lab. And then when I get them, so from that point onward, so for me to isolate the RNA, that takes me 3.5 hours. Then I switch and go to a completely different lab. And then I set up my RT-qPCR. That takes me about another hour and a half to pipette into all those little bitty tubes, little wells. And then the machine takes an hour and a half. So we're at about 6.5 hours so far without any breaks. So I do like to break for lunch sometimes. I actually usually don't, but and then when I get these results, it takes me maybe another hour at the most to go to this I'll show you in a moment the final results that I I pass on to Guy and to Gillian Ice, etc. I'll get to that in a moment. One thing, this method that I'm doing, I want it to be quantitative. So what I have to do, a bunch of these wells are actually controls, standard controls that I know exactly how much RNA I'm putting into them. And that's what this picture shows over here. It shows my standard curves, and I get some numbers from here, like how well are things working. This would be like really high concentrations of my standard. This would be really dilute, and you see it gets a little more spread out. And these X's, you see them kind of as slashes. These are actually these samples. So coming from this wastewater, you can see they're lying here on the standard curve. So this is the BCOV standard curve, and then the other three are like right on top of each other. So there's lots of controls built in, and I have to look through everything and make sure. There's a lot of quality control I have to do to check before I can then go to this point. So I take all that data. All of that data gives me like one little point here. So I graph them. What I'm showing you here is graphing. So the average number of average gene copies per liter of wastewater. So I have to do a lot of calculations, but I'm assaying two viral genes. So the N1, well, this one gene, the N gene, but in two different places. So N1 and N2. And as you can tell, they're actually lying pretty much right on top of each other. So that was really good for me. That showed me that they're kind of a control for each other. If I see a signal in one, I'm seeing a signal with the other one as well. So that lets me know things are working well. And we started this back January 14th. I think classes started the 18th or the 19th. So we were trying to get a sample or two in before the students return. But that's kind of a catch-22, because you need students in the buildings for the water to flow, for Guy and his team to collect. But then you've got students in the dorms that could already be bringing a positive signal. So we called these our baseline signals, but we're not, what does that really mean? What we're looking for, though, is not so much the number. It's kind of useful, but we're looking more for trends. And so we're looking to see what are these signals doing across time. And so, for instance, it took me a few weeks to get all the kinks worked out. So it was actually Super Bowl weekend. I think that was February 7th, 8th, Super Bowl weekend, when I finally got, everything was working beautifully and everything was all coming together. I was able to process all the back samples and everything. And I gave these results to Gillian Ice. She's overseeing I forget what her title is.
COVID operations is what she oversees for the university.
So she oversees like the students doing the testing, the clinical testing, and I'll tell you a little bit more about that in a moment. But I sent it to her, and what had happened at that point was right here. I had data up to here, and I sent her these, and I saw this, and I saw this big jump here, and I said, Gillian, you might want to check this out. And so she took a look at it, and she decided, she said, let's act on this. Call the students. I'm going to call the students in from that dorm. This one is actually just a single dorm. I'm going to call those students in, and I'm going to test them, our quick way of testing. And I'll show you that in just a moment, talk to you just a little bit about how that works. So one other time was here when this went up pretty drastically. Now, yes, there were things back here, but we didn't have it quite in place at that time, but now we do, and we saw this. And so the same thing, she contacted the Carr and Tanaka students and said, hey, we're seeing this uptick. Can you please come in and do this rapid testing just to see if we can find some positives that are sneaking through or something. So that's
Karen, I just want to point out something on your graph, and please correct me if I'm wrong, but that's a logarithmic scale, right? So the
That's
distance between the bars is not 1, 2, 3. It's actually 10, 100, 1,000, 10,000.
correct.
So it's a tenfold difference between each of the lines. So the difference really is significant for some of these changes.
Yes. So that's what alerted us here. Like this was nearly 100-fold. This was nearly 100-fold. That was what was alarming us and saying, hey, we still don't know quite what all this means. So one thing that we met this morning actually to talk about how can we put the clinical testing results together with our wastewater testing results, because someday would we be able to use the wastewater surveillance to give us an idea of what's going on, and maybe we can slow down on some of that clinical testing because that's expensive, and as Guy mentioned, it's hard on everybody that they have to keep going and getting tested and everything. So I just wanted to point out that OSU has been doing the Athens City wastewater. So they get a sample twice a week from the treatment plant, the Athens City treatment plant. And these are some of their results in red. This is showing the signal. They're just looking at one gene. They're looking at the N gene, but just once. And so you can see we're seeing some ups and downs. We're seeing some ups and downs over here. The black line are the clinical cases. So the known clinical cases over a 7-day average. And so we want to do a similar thing with our data, with the OU, the dorm data, and see if we can figure out trends and such. We're not there yet, but right now we think the wastewater surveillance is it's another tool. So because we are so robust in the clinical testing, it's not quite as important, but it could be down the road. And so just wanted to show that there's actually dashboards, multiple actually, but we are on this COVID-19 POOPS dashboard, and this is us right here. And there's both the university, and if you go to the second page, there's the city results. And not only that, this shows worldwide. So wastewater surveillance is going on everywhere. And we're still learning what can it tell us, but what we're hoping is that eventually we can use it, maybe slow down on the clinical testing, keep the wastewater surveillance going on so that, especially as people get vaccinated, the incidence, it's going to go down, but there's going to be regions or pockets of people, places around the world even, that maybe don't have access to that vaccine or don't want the vaccine. So this could be a way of kind of tracking to see if there's disease in the area, infection in the area. And I'm also wondering if it could be used, like, we don't know how long these vaccines are going to last. Like, will we have to do it yearly, like the flu vaccine, or will it be longer term? So I think this wastewater surveillance could also help there, monitoring like a whole city or a portion of a city or something, and just seeing, are we seeing prevalence of this virus?
Karen, you have a question.
Okay.
So before you were talking about one of the dorms and how you began to see the uptick in the wastewater. And then you said that the university decided to do the rapid testing on the individuals in that dorm. Was there a correlation between the numbers? Did we see an increase?
Well, what Gillian told me is that actually it wasn't through the rapid testing, but the day after I told her about that uptick, someone got their results back from the one of the methods that I'll mention in just a moment, one of the testing methods, the weekly testing method, and there was at least one positive person in that dorm at that time. And so in the wastewater surveillance, what we're going to possibly see is we might see it swing up as there's infectious people in there, but then the university policy is that if they identify positive people, those people get pulled out and moved to a quarantine dorm. And so the signal should come back down. So we're probably going to be seeing these swings somewhat, and that's why we're trying to figure out what do these trends mean, and can we correlate them with cases? We'll never be able to correlate it one for one, like this number means this many people are infected. That will never happen because everybody reacts a little bit differently to the virus. You could be exposed and your body can actually have different levels of the virus. And you can be shedding in your feces different amounts. I mean, everybody's going to be different. So that's why we're looking more at trends and populations. Okay, so on this slide, I wanted these last two slides, this one and the next one, I just wanted to talk just a moment, just tell you a little bit about how we can detect the virus in us, clinically. So one of the methods, we call it a molecular method, and that's basically very similar to what I'm doing with the wastewater. So we're looking for the sequence, the RNA signature, the RNA sequence. So we call that a molecular method. And that's actually both of the methods that are being used on campus is a molecular method. So one, the VALT method, VALT is the company, and the students are actually spitting into a tube, so they're collecting saliva, and then that gets sent to VALT, and VALT is doing essentially what I'm doing, the reverse transcription quantitative PCR, and that's the gold standard actually for detecting it. It's the most sensitive. So VALT is doing that and then returning those results in about 48 hours, give or take. There's a second method that we're using on campus, what we're calling the rapid test. That's by Abbott, and it's called ID Now. And it also is detecting the RNA, but it's not in the same, it's not doing the cycling. I didn't even talk about that, but it's a little bit different method than what I'm doing for my reverse transcription quantitative PCR. It also is quantitative in a sense, not quite in the same way, but it's giving a signal based on whether it sees RNA. There are also two other methods, types. There's a method that can look for protein from the virus. So for instance, that spike protein, and that would be coming from blood. That can come from RNA, saliva, nasal swabs, all those methods, that can come that way. And then there's a last one that's looking at for antibody production. This is actually the least sensitive because we make antibodies for all different reasons, but they're looking for an uptick of some of the antibodies, and that would be coming from the blood. And these methods are fairly quick, but they've managed to get some of the molecular assays to be pretty quick as well. These are a little less specific, and as I said, the RNA is the gold standard. And then finally, just wanted to show you, just mention a little bit, because the buzz right now is with the vaccines that are coming out. And so two of them have already been approved for use in the United States, the Pfizer one and the Moderna one. And everyone's been hearing, oh, they're based on the RNA technology. And so basically what is being done is the mRNA, just a region, just a piece of it, not the whole thing, not the whole viral RNA, just a little part of it, a little part of actually the gene that encodes the spike protein, is essentially purified and then encapsulated in lipid, in fat, and then that is injected. And then that, because of that fat layer, that can actually meld in with the cell membrane, which is also made of lipid, made of fat, and that can then release the RNA into our cells and can help us mount, well, first our machinery, our own machinery will make that spike protein, but just that spike protein, nothing else. So it's not infectious, it's not contagious, anything like that. But then that triggers an immune response in the body. And so that's the method for the mRNA, and it's been very, very effective. There's two other methods. So the next one, that's this Oxford AstraZeneca one, is already approved in the UK, it probably won't be approved for use until at least April in the United States. But there's a Johnson & Johnson version that I believe I think they did presentations today, and so this could be approved like within the next few days. This one, they're calling it the viral vector method. So again, it's the spike protein. What they've done is they've created the gene for the spike protein. So the virus encodes the spike protein through RNA, but these companies have kind of reverse engineered it and turned that into DNA. And DNA is a little more stable. And they then package that inside of a virus, but not the SARS-CoV-2, a virus that we're much more familiar with. Actually, it's one that can cause the common cold, but they've engineered it so that it doesn't. And it's a weakened virus. It can't reproduce or anything, but it can carry that DNA into us. So again, injection, and then the virus sort of spits that RNA into our cells, and then our body once again produces that spike protein, and then our body mounts an immune response to it. So that's these two: the one that was created in the UK, and then the Johnson & Johnson one that probably will be approved probably in the next few days. And then the third method is protein-based. And so what that method is doing, they are taking actually the RNA or the gene, and they're producing this protein, the spike protein, in cell culture. So like in a tube or in a big vat, and purifying it. And then the protein itself gets directly injected. And then that mounts the immune system. And those two, the Novavax one, I think will probably be coming along maybe in April. I think they're hoping to be able to apply for approval in April. And then the other one is much farther behind, and it's probably not going to be until, they said, the second half of the year. So very late summer, fall, before they're looking for approval. But these have an advantage with the variants. You've probably been hearing about variants. Mm-hmm. And these have an advantage in that they can easily take the genes that encode those variants and produce those variant spike proteins and then inject them. And so people will be protected against those variants. It's a little easier with those.
So Karen, we have 1 minute. Yeah. And I have been working with you and Guy on this since late fall. And I would like for both of you to just chime in very quickly and to tell, what was it like to be in the midst of a pandemic where you were basically kicked out of your labs, you tried to get back into your labs, you find out that there's this new assay for wastewater. How did you come together? How did you get the PPE? How did you get the assays? What was that like as a scientist to try and deal with that hot mess all at once?
Hectic. But fun. I've loved this. Guy, I give all the credit to Guy for even putting this on my radar. I mean, I knew about this, but I didn't really know about the wastewater surveillance. That I hadn't been seeing that yet. I was thinking, oh, I can't contribute to this. And then Guy contacted me. And he said, hey, maybe we could do this. I can get the water. Can you help with the molecular assays? I'm like, let me look into this. We started this actually in the summer. So we've been working on this since the summer. It's been long.
Yeah. I mean, one of the things I just remember in talking with you guys, how do we get the PPE? How do we get access, into the manholes? Oh my gosh, we're running out of plates in order to run your assays because everybody else in the country is using these. It just seems to be one stumble after another that you guys have just handled so well. So on behalf of the university, thank you. I do have another question, perhaps our last question. It says, you mentioned a virus found in peppers as a quantitative control for your samples. What does the vault saliva test use as a control to make sure you did that they did the test correctly? Do you know what they're using?
As far as I know, they don't use, the clinical studies don't use an external control like that. They're using, I think it's an RNA polymerase, but for some reason I was thinking it's a viral one. And so that, you know, that's a good question because I'd never thought of it that way. And because I did look into setting up the saliva testing for OU as well. And yeah, Guy, I don't know if you realize, I think your camera is reversed. So.
Oh.
We don't
can't.
see you. We see your couch.
Interesting.
So, but I am not 100% sure on that.
Okay.
I need to get to that. Yeah.
Just the last thing that I just wanted to emphasize is, you know, it wasn't like there was a catalog and you guys could just go and pick something off the shelf. A lot of these protocols that you developed with your students in consultation with researchers around the country who seem to be really eager to help each other. I mean, you've helped other universities have helped us. Can you talk a little bit about that? We have 1 minute.
I want to give a shout out to Colorado State University. The people there have been phenomenal. They have a huge, they got started very early on this, and they have been so helpful. Like all hours of the night, I can send an email and say, help. And they have actually sent me reagents to help with. They've been phenomenal. And yes, wastewater surveillance is sort of the black sheep of the family. All resources are going towards clinical testing. So the wastewater surveillance, we've had to use kind of what's left over, what people aren't needing for the clinical testing. And someone phrased it as the wild, wild west. Everybody's got a different technique because you work with what you've got. And yeah, like my centrifuge, I got that from Guy. It's like, okay, I can do 8 samples. Okay. So yeah.
And
the auto samplers, Guy, you had those.
Yeah. And we should say that the governor's office has really been instrumental in this. Someone in the governor's office thought this was a great idea and made the resources available to do this statewide at any university that was interested. And they're the ones who really made this funding available. And like we said before, we're trying to put it online in the city of Athens. And the city is seeking money for that right now as well. So
hopefully. Yeah, and at the university too, the CARES Act, the university had got money through the CARES Act. So that's what I actually used back in December to really get these assays finally going. So yeah, it's been phenomenal. And Roxanne has been great getting me PPE. So she was asking where the PPE is coming from. It's like, I don't know. Where's the PPE coming from, Roxanne?
They borrow and steal, I know. Well, I just wanted to thank you both so much for doing this cafe, and also just so much for just, you know, allowing students to work alongside with you. I think it's an amazing opportunity, and I think it's going to be really valuable for the university. So I think that's a wrap for tonight. Thank you.
all right. Thank you.
This was fun.